I will be talking to y'all about briefly about the journey of how I got to where I am, and also just a pretty cool story that I worked on in my graduate degree. And so I will say, and the previous class actually experienced this, I talk pretty quick and I talk a lot. So if you have any questions at any point in time, please slow me down or stop me and get my attention. So, okay. And also you could use the chat function if the unmuting is being weird. Okay. So, like I said, I'm currently in Denmark. I didn't know where it was until I moved here. But before this, I was actually in New York City doing my graduate degree, but I also did my bachelor's at Chapel Hill, North Carolina. But the place that I want to talk to you all about actually is this little island called Singapore. So a show of hands, has anyone heard of Singapore? Have not heard of Singapore? Possibly anyone's heard of them? Cool, cool, cool. I think some of you might have heard of it from the movie Crazy Rich Asians that was set in Singapore. So Singapore is a very small sunny island. It looks kind of like a sweet potato. It is summer all year round. It is 80 plus degrees all the time, which is glorious. But to give you a sense of how exactly small Singapore is actually 296 times smaller than the state that all of you are in right now. But somehow, and I'm willing to bet that Singapore has more people on its island than in Utah, because there are 7 million people, or the density at least must be much more than Utah. Okay, so growing up in Singapore is very interesting because it was warm all the time, I told you, but because it's warm all the time, life actually is able to sustain itself in a very diverse way. And so in Singapore, it is one of the richest biodiversity over there, and they're still finding new species every other day on that island. And so you can imagine if you grow up in a place where there's animals, insects, plants of all sorts, all colors, all forms, I naturally got very interested in biology. And so one of the core memories I had as a child was after a very long rainstorm. I was just walking around with my parents, and in the rain puddle, there were just these tadpoles that were just swimming about. And at first, I didn't believe my mom. I was like, that's not alive. There's no way. That's just dirt. It can't be. Like, where did these come from? And so we brought some tadpoles home and eventually made them, like, let them develop into frogs. Also, my parents are allergic to cats and dogs. So my most successful pet as a child was a mealworm, and it developed into a beetle and left me. I also had fish, but not very successful. These are betta fish, fighting fish, and all that. But I found myself actually getting very interested in photography because there was so much life in Singapore. And so here on the bottom right is actually a photograph that I took. It was just a moth that ended up flying onto my balcony, and I was intrigued by it, so I took a photo. Okay, so having life all around you really inspired me to ask a lot of questions about biology. And so growing up in Singapore, I had the great benefit of actually starting asking these questions at the young age of 12. And so the very, very first research experience I had was a very simple question. I asked whether or not used coffee grounds could be a good fertilizer. And so we selected this plant. It's an Ixora, which is an acidic plant. So therefore they like coffee grounds because that's also acidic. And we partnered with Starbucks. So any coffee grounds that they were done using, we just took it and we grew these plants with and without coffee grounds and measured whether or not it helped with the growth. And then when I was 13, I started a project on whether or not ants would be killed by garlic, because it's a kind of little known fact that garlic is a natural pesticide, but I was very intrigued in how the ant brain changed when they were fed garlic. And so what's really cool about ants is if you soak them in rubbing alcohol, so isopropyl alcohol, their exoskeleton becomes transparent. And so what I was doing was I fed ants garlic, threw them in rubbing alcohol, and threw them under a microscope and measured how their brain sizes changed over time. And so it was a pretty cool, but pretty straightforward scientific experiment. And then when I turned 15, I took a turn. I was like, I've done all this biology work. I'm sick of biology. Let's go do something entirely different. So I studied the effects of ferrofluids. And so the video that I'm showing here is a ferrofluid, which is basically magnetic filaments that are suspended in a liquid. And then when you put a magnet over this liquid, you see that this ferrofluid will align its shape according to the magnetic field. And so I thought it was pretty cool. That was initially why I joined. But then after a year working with ferrofluids, I realized I like studying things that grow. Like you can study the physics of this as much as you want, but I like seeing things develop. And so all of these years of just kind of like science fair projects and like unofficial or more simplified science projects led me eventually to my graduate degree, where I studied microbiology. And so microbiology is defined as the study of very, very small things, micro. And specifically, it's things that you cannot see without a microscope. So before I move on, do any of you have any questions or queries about what I've said so far? Because I've just rambled on for quite a long time. And a warning, if you don't ask questions, I'm about to ask you questions. So either way, you'll have to speak up. Nothing? Okay, cool. Then I will throw.
Hold on just a sec. I think one person's typing it into the chat. There you go.
Let's see. Question. Okay. What is the strangest animal you've been able to see in person? Strangest animal. I think that platypuses don't make sense. Like every definition of a mammal is defined by a platypus. You know, like a mammal shouldn't lay eggs, but the platypus is just out here being like, you know what? I'm gonna lay eggs. Okay. So I think it's pretty strange, but I'm trying to think what other strange things this could be. Microscopic worms are strange because they divide and they grow on their own. Okay, why does rubbing alcohol make ant exoskeletons transparent? That's a good question. I, as a child, didn't ask this question. I just was like, oh, great, it happens. But I'm sure there's some chemical explanation for that. I know that ethanol is a clearing agent, so it's able to take out pigment. It's the same as if you use acetone for nail polish removing. So it's a solvent that takes out pigments. And so I imagine that's something similar to rubbing alcohol to the ant. Okay, any more questions? Do you have any photos of what happened to the ants? Okay, this was done, I believe it was on a thumb drive that I lost a long time ago, or else I would have had photos. But they're just pretty cool. Okay, let me see if I can Google. Give me a second since we're here. Ant brain. Oh, they're not being helpful. Google's not helping. Okay, I apologize. Couldn't find one. But they're kind of small. They're kind of intriguing. Yeah. Okay, so the question I have for you all is, what is the very smallest thing you can see with your eyes? You can sound off in the chat. You can sound off however. Human hair. It's good answer. Any more guesses? Grain of sugar. That's pretty small as well. Yes, yes, yes. Grain of sand. I'm seeing someone say light, which is also an interesting answer. Dust, salt, dust particles, spiderweb. Spiderweb is an answer too. Sand, dust, 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 dust. Cool. So these are all very good answers. Actually, flower is an interesting answer as well. Okay. So the classmates that are saying photon and like microscopic things, that I don't think you can perceive with your own eyes. Like I don't think you've ever seen like a singular photon. But I will say one thing. When I found out that gravity works on photons, my mind was blown because somehow the light beam follows gravity. Just hold on to that information. It might not be helpful to you. Anyways, the answers that y'all have given are all really good. The answer that I found online is actually pollen. And so I don't think y'all said that. But so pollen is, in spring when you're like out and about and then you're using your phone, all of a sudden there's a yellow film on your phone. That's pollen. Or sometimes you see it all over cars or on the floor. Those are considered one of the smallest things that you can see with your eyes. And so what I have on my slide is actually a explanation of scale. So when you go from left to right, the things go smaller and smaller and smaller. So a lot of your classmates said human hair is pretty small. That's about 60 microns in diameter. But a pollen, this blue thing, do you see this blue little dot? It is 6 times even smaller than human hair. And so bacteria, which is what I study, that's a quarter of the diameter of pollen. And so from my knowledge, unless it's a very special bacterium, you usually can't see it without a microscope. And so I usually study, my subject matter is using a microscope. And if you go even further, viruses are even smaller than bacteria. So my study involves things that are really, really small that you can't see just willy-nilly. Okay, so my next question for y'all is, where on your body do you think bacteria live? Where do we have the most number of bacteria on our body? Thoughts? Anyone? Guesses are fine. Face, liver, mouth, nose, hands, all great answers. So far, most of the answers rely on the skin, which is a good observation because if you don't wash your hands, you'll have bacteria. Nose is actually an interesting answer because in our noses, there's actually 50% of us are likely a carrier of a very, very nasty bacterium. And that nose bacterium sometimes goes into your body and kills you. So skin, also, all that type of thing. But the actual answer, I actually saw it in the chat. Those of you that said stomach or in the gut, this is exactly where you have the most bacteria. And these bacteria are actually necessary for us to absorb all the nutrients from our food. And there has been some math done that they believe that there are more number of bacteria cells in your body than there are human cells. So we're made up of more bacteria than we are of ourselves, which is interesting to think about. But that number also varies based on your bowel movements. So for example, if you go to the bathroom and you have bowel movement, the number of bacteria in your body will drop very drastically. Cool. So all great answers. So bacteria don't only live on our body, they also are out in the wild. So those of you who have been to Yellowstone Park, if you see those geothermal vents, so it's very, very hot water, oftentimes you'll see these very, very colorful bacterial mats. And so these things are made up of billions, and trillions of bacteria that they're so dense together and they make this colorful pigments. And also, bacteria oftentimes grow in pipes. So in a hospital and a brewing setting, they often form along the pipes and then form nasty things and changes the taste of your beer. And also, for those of you who forget to brush your teeth, you might notice that a film starts to grow on your teeth, and those are all bacteria. So please, please don't forget to brush your teeth. Okay. And so that is bacteria on the body and also in the wild. But I told you all I'm a microbiologist, and we need a way to study bacteria in a lab setting. And so what we do is we grow these bacteria on agar plates. So it's just plates with food. So it's like a buffet type thing for them. And then they grow over time. And you can see they form these very dramatic and very beautiful patterns. And somehow they're able to do this on their own. Like they're able to communicate among each other and then form these structures. And so the bacterium that I study, one of my favorites, is on the bottom right. So it's red and super wrinkly. And this is made by a bacterium called Pseudomonas aeruginosa. It's a very long word. It is most famous for being the #1 pathogen. So the most nasty bacteria against individuals that are immunocompromised or individuals who have cystic fibrosis. On top of that, the Pseudomonas aeruginosa is also very successful at taking over all sorts of animals, plants, fish, cow, etc. And it is able to do this because it grows very readily and it grows into these communities very, very quickly. So my research in New York was actually studying quite literally a weapon that Pseudomonas made. And so this is how the weapon looks like. So they're called R bodies, refractile bodies that are colored in blue. These very, very coiled structures. Pseudomonas aeruginosa uses this against its opponents. And so I really think this R body is really intriguing, and I can tell you why. So the R body exists in two forms. One form is a very coiled structure that fits very nicely inside the bacterial cell. But when it's outside of the cell, it's able to hyperextend to 20 times its normal size. And so if it's 20 times its normal size, it can't fit into the bacterial cell anymore. And so my project was basically asking why would this small little bacteria make such a large sword? And also it just a lot about this sword defies physics, defies logic. But I'm probably going to age myself. But when I started studying this this sword, it reminded me of plastic lightsabers, which I don't think your generation plays with anymore. Y'all have better things to do. But it's pretty cool because here's a video of these R bodies. And so these swords, each black dot is a singular sword. And I basically subjected it to different buffer washes, and you can see the sword hyperextends and then coils back again, hyperextends, coils back again. And I was basically studying what is the point of this mechanism. Cool. Okay, so one of y'all asked me what my weirdest animal that I've seen in the wild. I gotta say this worm that I studied is pretty weird because we grow them on plates and they're also microscopic. They're able to just divide on their own. So quite literally, don't need no man, and they're able to divide. And so I use these worms as a host. So I put my bacterium into the worm and see how they reacted over time. But on top of that, I also put the bacterium into plants and see how the plants fared over time. Okay, so that was kind of what I was studying. Quick show of hands. Who of you owns glow-in-the-dark toys? Is it just me? No, is glow-in-the-dark out of fashion already? So I'm almost embarrassed to admit this. I, as a child, had so many glow-in-the-dark stars in my room because I was afraid of the dark, and so I really made use of this technology. But then I was thrilled to find out that in biology we use something similar. So what we're able to do is we take these bacteria and we tell them, hey, I want to see where you are. So we give them a protein that glows in the dark. And so what happens is when I feed the worm that's transparent, the bacteria, I can look at where the bacteria ends up over time. And so here is a not so happy camper of a worm because this worm has eaten this nasty bacteria. But what I hope you can see is every single one of this red color is the glow-in-the-dark bacterium. And on top of that, if you zoom in, you start to see this worm has a lot of bacteria, but not all of them are red. And so that was another research question I had of why only a small part of this population was red. But that was pretty cool. And another thing I did was also I was able to make glow-in-the-dark plants, sort of. And so on the left is just an underside of a leaf, and you can see all the mouth holes of the plant. And then on the right is the glow-in-the-dark plant. So yeah. So the last thing I want to say is this is a picture of all the people that have really helped me through this specific lightsaber project. And I hope that y'all can take away that science is not a solo sport. Science has never been a solo sport. That no one person is able to solve all the scientific problems on their own. That a lot of times you are not the expert in a certain field. And the good thing about science is you can go consult the experts. And so I am not here to, I would not be here today if I didn't have the support of people that have taught me before. But I'm also, the reason why I moved to Copenhagen is I'm now in a multi-international collaborative effort with 13 different countries studying the effects of bacteria on chicken, for example. And so just wanted to throw it out there that I wouldn't be here without the support of a lot of people. And with that, I would like to thank you all for your attention and take any questions. Okay, cool. So I think the last period, it was just a free fire of questions, and then I just scrolled through it and slowly answered all of them. So feel free to do that, and I'll try and get to all of them because we still have quite some time. So how do the bioluminescence work in some parts of the ocean? That is actually a very good question, because the way we make bacteria and plants glow in the dark is we actually borrowed this bioluminescent protein from the jellyfish. So the jellyfish are very, very good at making bioluminescence. And so we took that protein, put it into plants, put it into bacteria, put it into worms and human cells, and that's how we got bioluminescence to work. So it's actually a very, very good tool. So I would say the jellyfish have better ideas than we do about bioluminescence. It's interesting that everyone keeps asking me about how much money I make. I'm not going to do that conversion because it's in euros, so I apologize. What makes it so that the glow-in-the-dark stars can glow? So that is interesting because the abiotic glow-in-the-dark is something about you need photons to charge up the material that's in the glow-in-the-dark. They go to a higher activation state, and then it comes down and releases a photon. So that's the abiotic glow-in-the-dark. What was your most interesting discovery? That's an interesting question. I haven't thought about that. I think we didn't expect these swords to be in the bacteria. Because if you think about it, bacteria is this big and the sword is this big. Why would the bacteria make such a big sword for no reason? So I thought that was pretty cool. And just I like pretty images, photography. So that was a pretty cool topic. Do you travel around a lot? I have been traveling a bit more here because I'm working with 13 other countries. And so in March, I'm going to Barcelona to collaborate with some people. Okay. What type of education do you need to become a scientist? So that's interesting. So this might not answer your question, but I feel like what defines a scientist, that you sitting in your desks right now could very well be scientists already, because a lot of times we apply the scientific method without even realizing that sometimes when you decide that, oh, which brand of milk do I like more? You go about this in a scientific method. And so does that make you a scientist? I think so. Okay. What was your most interesting? Same question. What do you say most revolutionary discovery in science ever. I'm biased. So I really like genetics. So to me, when we figured out how to modify DNA in our host models, so like the mouse, the worm, Drosophila, plants, I think the ability to ask these type of questions with genetics is very important. Because if you, for example, my project, I was studying these swords. So I had one bacterium that made the swords. I had one bacterium that couldn't make the swords. So I took out the genes from that bacterium so it wasn't able to do that. And so I think science has progressed a lot now that we're able to do genetic engineering in those host models. Okay, let's see. Who's your favorite scientist? I just realized someone asked me about Bill Nye just now and I forgot to answer. Who's my favorite scientist? I believe there are a lot of women scientists that have done so much work that because of misogyny in the olden day, people just take their results and not credit them. So the discovery of DNA was actually discovered by a woman, but it's often credited to these two other scientists instead. And so, yeah, I hope that answers the question. Have you ever blown up a science project? This is an interesting question. Blown up. I don't think so. I think the previous section asked also, and I said I put dry ice into tubes and then that explodes, but don't do that. That's not safe. How many years of school did you do? Okay, oof, let's see. Went to college, then I did 6 years of PhD. So in totality, my brain's not working in math. So I was in school from 7 till 28. That's a lot of school, but less school than doctorates, or not doctorates, than actual doctors. So I have friends who started with me as MDs and they're still studying. Okay, let's see. What motivated you to pursue your passion in biology? I feel like that was just the topic that resonated the most with me. It was also my best subject growing up. But then it becomes a chicken and an egg of, did I do well because I liked it or I liked it because I did well? I'm not sure, you know. But I was telling the other class that I was really bad at math. I didn't get physics in any way, shape, or form. Literature is beyond me, but so it kind of made sense for me to go into biology. Okay, have you done research on mutations? And if yes, what was your favorite thing you learned? If not, what do you want to know? That's an interesting question because I think most of my projects from college onwards were mutation related. I either introduced a mutation or I took out genes, which I think is also mutations. Another thing with bacteria is I did a genetic screen. I don't know if this is too much detail for you all, but it's basically I threw a junk piece of DNA at the bacterium. It would jump into random parts of its genome. And depending on where it jumped into, I could see how the bacteria changed. And so that is a very classical mutagenesis study that you would read in textbooks. Okay. Mutations. If you couldn't do biology, what would be your second choice? Telling the other class that I'm actually certified to teach 9 through 12 high school biology. And I honestly think if I weren't in the labs, I would be in the classroom. What is your favorite thing to study? Ooh, recently, I think transparent worms are pretty mind-blowing because you can just see what's happening inside the worm, and that's pretty amazing. So I'm gonna say transparent worm. How many experiments have you done? That's an interesting question. I think in the PhD, you end up doing a lot of experiments every day. And so the goal of the PhD is to amass enough experiments and tell the story with it. And so I don't think I could tell you an actual number for the number of experiments I've done, but it must be above 100 at this point, probably. Okay, if you had to be a different scientist other than biologist, what type of scientist would you be? I wish, okay, so there are two answers. I wish I was a computational person. I wish I knew any sort of coding, but I do not. That is not in my life, and that is not my affinity. I also think that chemistry is a more controlled biology, if that makes sense. So the chemists are often making fun of us because the variability in biological science is much larger than, say, making a compound. But part of me also wants to say like anthropologist, because who knows, right? Like the study of humans. Okay. When do you think scientists will dramatically change the DNA in humans? That's an interesting question because I feel like we've been doing it for years. So when I was, I think your age, I was studying about this method called the gene gun, where they quite literally took a gun, not a real gun. They took a machine and then would inject DNA at patients. And it's been shown to be helpful for different types of wounds, I believe. So I think we've been doing it for a long time. And widespread usage is hotly debated, but we've had these tools for years. Yeah. How did the pandemic? That's an interesting question. So during the pandemic in New York City, oh my goodness. So I was in the epicenter of the pandemic back then. And so it was just a weekend. And I was like, okay, go visit my friend. I'll bring my laptop just in case something happens. Nothing's going to happen. It's not. So I went, and then we had a total lockdown. And they were like, yeah, it's just gonna be 2 weeks. 3 months later, I'm still in lockdown. But what's pretty cool is that at the time I had done enough experiments that I could spend 3 months doing data analysis. I learned how to make phylogenetic trees. I pivoted into more, they call it in silico, which is just computer-based research questions. And I wish I had a larger computational brain to do more in-depth studies, but I learned a little and I did some of that. Yeah. Okay, let's see. But I will say, post-pandemic, I think scientists realize that we aren't talking to each other enough. That like we have the power of Zoom. We have the power of multi-international studies. And so I have to say collaborations in science, I believe, increased after the pandemic a lot of folds. Yeah. Okay. Have you ever studied an albino animal's mutation? I have not personally done it, but usually it's just a mutation in the melanin producing pathway. And so if you can't make melanin, you'll end up being albino. Okay, can you change my DNA? I will say this. I study bacteria, plants, and worms because I do not care about the human body. If I cared a little bit about humans, I would be a doctor, I believe, like a medical doctor. So I personally would not change your DNA. Well, the dinosaurs come back with DNA. I was just in a natural history museum recently, and I don't think we realize time in the dinosaur age entirely different. If you think about it, because the distance between the sun and us were different back then. So a day in dinosaur day, entirely different from us. Climate, entirely different. Bacteria makeup, also entirely different. So as much as we try to be like, oh, we can bring back Jurassic World, I think the ecosystem has changed so dramatically differently that I don't think dinosaurs can be sustainable anymore. Yeah, because I learned so many things in this museum. The fish were unbothered. So the meteorite killed all the dinosaurs. Fish, meanwhile, were just like, we're in the sea. We're entirely fine. Fish are the cockroaches of the sea. Okay, let's see. If a heart transplant is possible, do you think it? Again, I am going to defer to, I don't care about humans. I apologize. Where has been your favorite place to study? I feel like my time at North Carolina Chapel Hill was the most enriching in that the school spirit around Chapel Hill was just great. And I think the mentors I had back in Chapel Hill were also the most supportive of me because we were studying CRISPR-Cas in the plant. Okay, let's see. Is there dinosaur DNA? What is there dinosaur DNA inside of sap? I don't think I understand that question. What's sap? Plant sap?
It's like from Jurassic Park. That's how they found the DNA was inside sap that then turned to amber, and they like go and get a mosquito out of it.
I see. Okay. I don't know. I don't know. I am though. Okay. I'm currently in the Natural History Museum of Copenhagen. And so they are, so if anyone would know this answer, not me, someone else in this building, they study ancient DNA. And the things that they do is mind blowing to me. They get DNA from a dead mammoth. They get bacteria DNA from mummy corpses. I don't know how. And so the questions that the ancient DNA people are asking are beyond me. Yeah. Do you have a Valentine? I hate to be this person, but I actually don't care about Valentine's Day. I feel like we should celebrate our loved ones every day. Why is a dead white person the reason we celebrate love? So anyways, I do not have Valentine with me right now. Are we monkeys? No, different. We've diverged so aggressively differently. I also learned in the museum the other day that monkeys have a much smaller brain capacity than we do. So if you just look at our skulls are way bigger. Okay, if you know, at what point does something die, or rather, how do things die of age? Okay, I have two answers. At one point, does something die is interesting to me because in the humans, I think we believe when our heart stops is when we call time of death. But then one would argue that the decomposition process is another form of life because as your body decomposes, bacteria continues to grow, fungi continue to grow. Other types of things, other forms of life are using your dead body to continue to grow. And so to me, I don't know. I don't know where life ends and starts. And how do things age? I have a very molecular answer to this that might not be of anyone's interest. Scientists have shown that as we age, the length of our telomeres, so the length of a specific DNA sequence in our body becomes shorter. And so when they cloned Dolly the sheep, I hope you'll have heard of Dolly the sheep, Dolly actually died halfway. So Dolly's sheep died way earlier than scientists expected because they think that the telomeres that she came from was already truncated. So she didn't have that much time to live anyways because her DNA was shorter. So yeah, that's about aging. But lobsters do not have telomerases. So lobsters are invincible. Lobsters just keep growing. If you give them enough nutrients, they'll just keep growing. Okay. Can you make a megalodon? I personally cannot. I apologize. What is the strangest mutation we have ever? Oh no. I don't know if we want to go down this road. So the worms that I studied, they're hermaphrodites, which means that they're able to self-fertilize, make eggs, and then lay eggs and continue. One of the labs that I worked in was studying the vulval phenotype. So they're able to make mutants where the worm is not able to give birth to the eggs. So what happens is this worm will fertilize the eggs will hatch inside the mother, but then the babies just eat their ways out. But so in terms of genetics, you still get a generation. And so the geneticists were like, great, let's move on with our lives. But you can imagine sometimes in the microscope, you'll just see quite literally a bag of worms. And that's the phenotype that we call it. You asked the question, I do not apologize. Okay, are there still megalodons? I don't think so. Can I cure cancer? Absolutely not. Also, I will say, Curing cancer is a very complex thing because every person's cancer is different, and that's usually why it's so hard to get rid of.
Okay, Brian, thank you so much. That was amazing. Thank you for answering all the questions. It's such a good example for my students to see what a real scientist is like. Thank you again for last class period and this class period. Thank you.
Thank you all so much for asking all these questions too, because sometimes I've done this before where people just stare at me and I stare at them. So it really helps when you all are engaged and ask these type of questions. And so yeah, thank you so much for listening and asking all these questions. Yeah.
Thanks, Brian.
Thank you.
Thank you.
Thank you.
Brian.
Thank you.
Thank you, Brian.